I could interrupt all these fabulous conversations which are taking place in the room. Thank you all for coming, and I want to extend a welcome to all of you on behalf of Gordon College and also the Center for Faith and Inquiry. My name is Paul Brink, and one of my roles here is as the director of the Center for Faith and Inquiry, working with Ryan Soltero, who is the program director. It's so good to see you all. Thank you all for coming. In a few moments, I'm going to invite Craig Story up here to introduce our speaker. First, I'd just like to take a few moments to share a little bit with you about our lecturer's namesake. It's Dr. Robert Herrmann. Dr. Herrmann was a former adjunct professor here and pre-med advisor at Gordon. And he also taught medical school biochemistry for 22 years at BU and later at Oral Roberts University. But in 1981, he left medical education to become the executive director of the American Scientific Affiliation, which I know many of you are familiar with. It's over 2,000 members, society professionals who are interested in questions about the integration of faith and the natural sciences. Gordon College has an active ASA student chapter. Many of you may remember the ASA annual meeting, which was held here, I don't actually remember when, 2018 about, or thereabouts. So, as part of Dr. Herrmann's work with the ASA, he met Sir John Templeton, and the two collaborated on several books, and in fact, worked together to help found the John Templeton Foundation. And today, we're very grateful that that very same Templeton Foundation serves as a key sponsor of this whole lecture series. And so in a very indirect way, Dr. Herrmann arranged for the funding of his own lecture series, which is very clever. I'd like to also welcome Lisa Spencer here, but I will allow Craig to provide a more formal introduction.
All right. Thank you, Paul, and again, thanks everyone for being here. My name is Craig Story from the biology department here at Gordon College. Worms, wheezes, and new weird diseases, depending on what you're looking at. Great title. Dr. Lisa Spencer, PhD, is currently the scientific director for the Gastrointestinal Eosinophilic Diseases Program at Children's Hospital in Colorado, Denver. That is indeed a mouthful. But in plain English, I can tell you that her years of studying the eosinophil, EOs, as they're maybe known, is a special type of immune white blood cell. This led her to be recruited there in 2018 from the lab she established at Beth Israel Deaconess in Boston. Dr. Spencer is one of the world experts in the biology and behavior of these kinds of cells, which previously were only known to help fight worm infections. And now we have an appreciation of a much broader role of EOs in several chronic conditions, which I think she will describe for us. What you won't know from reading her bio is that once upon a time, we both applied for the faculty job that I currently hold here at Gordon. Yep. So why did I get the job? We're both alumni of Gordon, but I graduated in '89. And she in 95, I believe. That's 6 or 7 years apart. So I had an unfair advantage. It's a near certainty that my having completed a postdoc gave me the edge. So I hope there are no hard feelings. And I don't think there are any between us. Dr. Spencer planned at one time to be a medical doctor, ended up earning her PhD at UConn Medical School and doing her postdoc at Beth Israel, her own postdoc. More recently, she's hosted several of our own pre-med students in her lab for internships and collaborated with Dr. Angie Cornwell and other Gordon students on some really interesting projects. But certainly that decision of who Gordon would hire back in 2002 really did have a big influence on our respective career paths. And I can safely say and confidently say that Dr. Spencer has made and is making great, widely recognized contributions to the world of science with many significant and publications that are highly cited by researchers in the field, with elections to boards and societies and all the good stuff. So it all seems to have worked out in the end. So let me say thank you, Dr. Spencer, for coming, share with us here and in some different classes this week, and welcome back to Gordon.
Thank you. I was a little worried about what Craig was going to say. That was pretty tame. So we're good. You made the right decision. This was, I would agree. There's no hard feelings. A little bit of counseling in there, but no hard feelings. So I can't take full credit for this title. I did kind of steal it, I'm going to be honest, with permission. Peter Weller, who is one of my mentors over the years, titled the lecture this, and I loved it. And so I asked if I could borrow it, and he said yes. So there you go. So thank you so much for having me here. It is really good to be back at Gordon. This is just obviously a very special place. So I appreciate being here. And thank you, Paul, and thank you, Ryan, and the amount of work that goes into something like this, all the organization. I really appreciate that time. And the Templeton Foundation and all of the work that's been going on to have a forum like this is really unique. And so just being outside of these walls, I think you forget how harsh it can be out there. This is a bubble. It's a wonderful bubble, but it can really be a bubble. So the fact that these forums exist is really powerful and important. And so thank you for keeping that going. I do have one story about Dr. Herrmann. He was on kind of leaving some of his responsibilities here when I started as a pre-med student. So I didn't have a lot of interactions with him. But I did have a meeting before he was putting together my composite letter where he kept talking about me as a pediatrician. And I kept changing his wording and saying, no, infectious diseases, internal medicine, adult medicine, definitely not kids, they're sticky. And it's not lost on me that I now am in a department of pediatrics and working in a children's diseases. So he was prophetic among all of his other talents. So why research? As Craig alluded to, I came here as a pre-med student, and I put my head down, and I buckled through, and I got my grades, and I did well in the MCATs, and I was all ready to go. And then I started thinking about what life was going to be like and all the patient care, and I realized the part of medicine that I liked the least was the direct patient care part, and that was a huge red flag for dropping hundreds of thousands of dollars to go to medical school. And so there are two people that I talked to about this, and they're actually both here, so sorry, guys. First, I went to Dr. Wright, and I said, I don't know what to do. And his advice, I'm paraphrasing, find what you love to do and do it. And don't be afraid to take a step along a path, then change your mind, turn around, and go do something else. So that was freeing for me, so thank you for that. And then there was Dr. Camp, who I said, hey, I don't know if I want to see patients and I'm pre-med, what do I do? And he looked at me, he goes, duh, you're a scientist. And there really was a duh in there. And so he saw it. And I didn't believe him at first, because you're looking at someone who really did not like labs at Gordon College. I am sorry. But the upper level ones, okay, but you have to learn skill sets. You have to learn how to ask a question. You have to learn the process, and that is just not fun. And it's not the same as asking a question, finding really innovative ways to answer it, and then that discovery moment when you find something brand new. Because in your lab, when you're titrating something, it's going to be titrated, or not if you've messed up your reagents, which also happens, but it's just not quite the same, and so don't let that be just discouragement to you. Why basic science, and what is basic science? I think some of us have seen this type of a graph before. If you look at industry, at some of the pharmaceutical pipelines, to get one medication FDA approved takes an incredibly long span of time, and it goes through many different iterative steps. You start with many, many compounds here. I listed 10,000. I made that number up. It's an average. We then whittle that down to fewer numbers of compounds in preclinical models. Often this will be a mouse model. Sometimes it'll be patient tissues. And then you finally maybe get a handful that make it into clinical trials, and there are 3 phases of that at least. And then eventually you get to FDA approval. That's all well and good, but one of the things that I was frustrated about with medicine, in addition to actually seeing patients, was that we had unanswered questions. And so before you can even come up with these 10 compounds, you have to find the pathways or the target or the basic concept that needs to be fixed. And so it's really that upstream piece that is an indefinite timeline. There's no clear, this is what I'll have achieved within 6 months to a year. It's really an open-ended, I'm going to ask questions, and I'm going to try to make the right question so I get an answer that's meaningful. But at the end of the day, we're asking a lot of questions and then following where those questions go. And if we don't do this, we don't fill that pipeline. And if we don't fill the pipeline, then we don't get the medications. And I think sometimes we forget that. And I think that's important whether you're in science or not, because if you're a taxpayer and talking about NIH funding, these are the types of things that we have to be aware of when we decide what kind of research to fund. And so there's some quotes from Lewis Thomas. He has a very cool paper out talking about medicine through the ages. And he was the chancellor at Sloan Kettering in the 80s. And this sums it up right here. Most of today's promising medical advances are results of fundamental research begun 20 or 30 years ago. Although my son, a few years ago, asked me what it was like to live through World War II, I'm not that old. So I have a few more decades before you should expect, the groundbreaking Nobel Prize winning research. But the second part of this sentence was equally important. It's by investigators with something very different in their mind. We don't see the end in the beginning. We don't know where we're going necessarily. It is really hard to write grants with this, by the way. But that's the reality of it, is often we're asking these questions. It's bringing us to a place where we didn't even know where we were going. I'm going to walk you through some of my journey with the eosinophil. It was a great introduction. I can skip some of my slides. So you're going to see in that story that we ask a question, we read God's blueprints to get the answers, and then that answer applies to many things that we hadn't even anticipated along the way. So this is how any of us can look at basic science, but I think Christians have a whole nother layer to this. So the way I look at science, what I think about every morning when I go into work is I am working here in a space as a conduit for future blessings that God is going to lay out. And so I don't have to know the end because God does. I can ask the questions in faith and know that he's bringing me to a place that is going to heal what he is wanting to heal. And so this becomes exciting because this means our God, who's outside of time, gets to direct where we go, and we can be part of that and participate in that even when we don't know where we're going. So how we approach this, I think the two big pieces for me are compassion. This is what prompts our questions. It's the least of these, and I think it's relevant right before Easter to talk about one of the messages Jesus gave, one of the final messages before the crucifixion. It's the whatever you did for one of the least of these brothers and sisters of mine, you did for me. And so this is what drives the question in science. And once you have that question, you're reading the blueprints. So this is, God has given us the answers to these questions. We get to read the blueprints there and then share it with other people who haven't learned that language and frankly have no desire to. So this is all part of the big picture. We're invited along this journey. And so I talk about reading God's blueprints. This is the same as Faraday's famous quote of thinking God's thoughts after him. So I hope you're going to see glimpses of this as we go through. This is how I approach basic science. It is inspired by compassion. It's continued through worship. And if we follow this faithful exploration, then we can be a conduit of God's blessings. Again, you don't know where that conduit is going to pour out, but we know it will. So where did we start in the journey? I came to Gordon. I was driven by compassion. I wanted to serve the least of these, and as every good Christian student from a Christian college, that meant you have to go overseas, and it is medical missions. So this is where I started. I was really looking for something that could relate to a medical missions career, and I found T.V. Rajan, and I will not pronounce his first name. He's asked us not to try because he's done hearing all of the massacres of his name. But this was an investigator at University of Connecticut School of Medicine, really brilliant pathologist, and he was studying a disease called lymphatic filariasis. So it's maybe more commonly known as elephantiasis. And if you can imagine the morbidity that is associated with, especially in these cultures, what this can mean for your ability to provide for your family and be a productive member of society. This is also one of the 20 neglected tropical diseases identified by the World Health Organization. So for me, in my, at that time, definition of the least of these, this is where I went. And this is not something you'll be tested on. Don't worry about all the words. It's carried by a mosquito. The infective stage is about 1 or 2 millimeters in size. The mosquito is going to bite, drop that little 1 or 2 millimeter squiggling worm on your skin. It's then going to crawl through your bite mark. Aren't you glad you just had cookies. It's going to climb through that bite, and it's going to go to your lymphatic system. So when we think about parasites, you think about the gut, right? So when you want to get rid of that parasite, you just end up passing it. You can't do that when the parasite's sitting in your lymphatic system. So we have this little 1 or 2 millimeter worm that is now going towards your lymphatics. In about a week and a half, it molts to an L4 stage. Now it's 3 to 6 millimeters in length, still manageable. But then it ends up 4 to 6 weeks later molting into an adult, and the adult females can be 8 to 10 centimeters in length. And so now we start to really have a problem, especially when they're 200 microns in diameter and your lymphatic vessels are about 10 to 15. So some people can clear these infections, some people get the chronic pathology, some people, the adult worms are able to live, produce their microfilaria, and then the whole cycle can repeat, and that's the majority. What we really wanted to know is, how are those patients who are clearing that infection very, very quickly, having no pathology, that must be killing those worms when they're only about 1 millimeter or 3 millimeters large, how are they doing that? And so we have a blueprint. We have endemic normals who live in an area who don't get infected. We also have permissive or non-permissive lab strains of mice who clear the infection just as well. And so we used a lot of different genetic mutations and a lot of different lines of mice to ask this question: how is it that an immune system can get rid of a big worm? This is a cross-section of the worm sitting next to one of our lymphocytes, just so you understand the magnitude of the problem of what this immune system has to deal with. So the approach is to use different lab strains of mice. We're going to find out what this pathway is. We're going to confirm those pieces are all in the human. And I am not going to walk you through all of the data, but I will give you the punchline. This is how you kill a worm. So this is an L4 stage larvae. It's about 2 to 3 millimeters in length. Those little dots are cells. And so lots of cells go and they stick to the worm and they just grab on, and they do it in a very coordinated fashion, and they actually encase it. So a worm has to pull all their nutrients through their cuticle. So when you cover the cuticle, they can't get their nutrients, they can't breathe, and eventually you immobilize them. So you basically form a granuloma. That's how you kill a worm. And here's the pathway of how you kill a worm, which again, we won't walk through in detail. There are a number of papers that we put out at this time. Or I have a 201-page dissertation, and I'm very happy to share that with anyone. It details this process. So this was what we were finding. And if you see this cell with the pink dots on it, that's an eosinophil. And it's sitting right above the parasite because, as Craig alluded to, eosinophils kill parasites. They dump some toxic proteins on it. So that all made sense. But what I'm not showing you is this process will occur whether it's an L3, an L4, or an adult worm. But if you really want to kill that L3 or L4, this has to happen quickly. And the only way it's going to happen quickly is if you get this little protein called IL-4 or interleukin-4, and remember that because it's kind of important. IL-4 has to be produced very early on, and it turns out eosinophils were the ones producing that IL-4. So all of a sudden, they showed up in some place they shouldn't be. So if they came in early and they dumped that IL-4, you got this really fast response, you cleared your infection, and all could go on. So I have to introduce you a little bit to this cell because this is when I met the cell that I then followed for the rest of my career. So here's an eosinophil. It's got tons and tons of these pink granules all over the cytoplasm. The nucleus is purple, and that's because we dump a dye called H&E on it. It's hematoxylin and eosin. And I have to say that because I had a 5th grader come up to me and say, it is so cool that we have purple centers in all of our cells. And I had to kind of shatter her illusions to say, no, we dumped the dye on it. This is what they look like without the pink dyes. So sorry about that. But so here is an eosinophil, actually one of mine, so it's pretty healthy. The big kind of open, opaque spaces in the center, that's the nucleus. All of those dark circles, this is a phase contrast image, all of those dark circles are granules. All of those granules have a bunch of different toxic proteins in them. So think about these as little bags in the cell full of biological grenades. So it makes sense that their whole role is going to be take these biological grenades and throw them at a parasite. And that's what my $150 immunology textbook from the 90s told me about eosinophils. That's it. I paraphrased a little bit. But so in this country where we don't have parasites, they're tended to be looked down on because you seem to get a similar response in something like asthma or allergies. And so the idea was, well, our eosinophils, we really need them for parasites, but they get a little confused when they see an allergen and they dump their granules and then you have tissue damage. So this was the reputation of the cell that I have now spent my entire career on. There you go. So again, when we see them upstream, that was new and that was exciting. So that's what I decided to follow, is how are they secreting that IL-4 and how is this all happening? If you remember, I made the comment, we ask a question and then we have an answer and the answer applies to lots of different things. We now know this pathway of eosinophil-derived IL-4 modulating your macrophages is important in many other diseases and in many other processes. So science, you don't know where you're going, and yet you end up there. So how do eosinophils secrete this IL-4, and how does that shape immunity? And so for these studies, I sought out, I was actually very lucky to be accepted into the lab of Peter Weller at Harvard. And so he is trained in parasitology. He's an expert in allergy and infectious diseases. He's a world-renowned hypereosinophilic syndromes expert. So he's an expert in this field and the top person, and he was asking the same question, which is also encouraging when you're starting your postdoc and you have a question and someone of that caliber is asking the same one. So we looked at these cells. For these studies where our human blueprint is the eosinophil itself, and I told you that they have these bags of biological grenades in the cell. Apparently, these bags also contain IL-4. So we took the granules out, we popped them open, we looked at what's inside. It's preformed and it's in the granule with those toxic molecules. It's not just IL-4. Other groups were also looking, and we compiled all of our data, and there's over 36 chemokines and cytokines preformed and stored in these granules. I didn't include them all here. I chose a few to include here, which are not important, but if you're an immunology student, you might notice those orange ones, all Th2 driving cytokines. So if you have an extracellular parasite or an extracellular bacteria, you need a type 2 immune response. If you have an intracellular bacteria or an intracellular virus, you want the things in blue up there. There's your COVID response. If you are pro-inflammatory, that's IL-6 and TNF-alpha. IL-10 is more of an immunoregulatory, and TGF-beta is a resolution. So we've got really diverse cytokines all packaged in these granules. So we wanted to understand, how on earth can you have all of these things packaged into the same granules? How do we sort that? How does the cell decide I'm going to pull this one out and send it? Because remember, if you grab your toxic molecules when you're trying to start a Th2 response, you blow up your tissue. And so that's a bad thing. So we looked into, we collaborated with Rosanna Mello. So she and I actually postdoced together in Dr. Weller's lab. And then she came back every year to Boston for several months. She's an expert cell biologist and electron microscopist. And she gave us a view of inside the granules. So she used electron tomography, which is electron microscopy on steroids. It allows you to do a three-dimensional image of what you're looking at that kind of a level. And what she showed us was full cities inside these granules. There are tubular structures that are dynamic, and they form, and they move around. And one of the papers I published during this time showed that we have receptors being expressed in those membranes. And you're reorganizing your cargo because your receptors in the membrane are being moved around, and they're grabbing their specific ligands. Anyone who hated studying form and function in the basic classes, these vesicles, do you see how they're just long tubes? You've got a really high surface area to content, luminal content ratio. That makes sense if you're having to hold all of your cargo on the membranes themselves. So for the first time, I'm not bored by form and function. It really makes sense in a biological situation. We also had some serendipitous findings, which are the best kind in science, right behind accidental. So here you have on the left, a healthy eosinophil. This is what they should look like. They're crawling around, they're happy, they're engaging with their matrix. When they go out of focus, that's because they decided to let go and float around a bit, and then they'll flatten out again and go. So that's what they should look like. We started noticing a lot of our cells under certain conditions didn't look like that. And so as any good PI, I told my technician they'd made a mistake and were killing the cells and do it again. And when they did it again and again and again and kept getting the same results, eventually, a little later than I should have, I realized it was real, along with some help from colleagues pointing out to me that it was real. This is what an eosinophil undergoing a specific regulated cell death pathway looks like. So you've got granules that are like shooting out to the left, and the nucleus just decondensed. And the non-scientific term for decondensed is splat. So the nucleus just splatted. You've got DNA in your nucleus, and anyone who has done a DNA extraction knows you get a gooey, gloppy mess. Well, DNA is sticky, and that becomes important because there are two consequences of etosis in the tissue in patients. One, those cell-free granules, we've shown they have receptors on the surface. They actually respond to stimulus, and they still function in the tissue. So the cell might be gone. The pathologist didn't count an eosinophil there, but if it's got scattered granules there, they're still functioning. They're still important in the pathology. Number two, you've got DNA traps that are sticky, really, really sticky. So those spider web-like structures that you see there are the DNA traps that got shot out of these eosinophils. And what you see captured in those DNA traps in the far left is Staph aureus. Those are those circles, so there's a bacteria. You've got the rods in that center image are E. coli. And on the right-hand side, it's Candida albicans, looking like little clusters of grapes. That's a fungal infection, important in immunocompromised patients. So here's a whole new function. Remember, this cell is only supposed to kill parasites and then be irritating to North Americans who don't have parasites. And we're seeing a whole nother world here. Shige Yuiki's picture is here because he is a physician from Japan who came to the lab to work with Peter for a number of years and collaborated on a lot of these studies. So again, we have a common theme, right? We ask a question, we discover something, and then we find out down the road it has lots of implications that we did not even anticipate. The same case here. So a number of different diseases, we now recognize this happens. I had Crohn's biopsies sent over, and we started slicing them up, and several of these patients will just be absolutely littered with cell-free granules. So there's so much more going on in the pathology here that we just didn't see before. We didn't know what the right question to ask was because we didn't have the information in the data. There are also two here that have an orange star. And that was, these are rare diseases. So hypereosinophilic syndromes is, I didn't study it specifically, so we won't get into the data, but I will show you a patient in a moment. An eosinophilic esophagitis, which we may not still call a rare disease soon when the diagnosis gets up to the level that we really are detecting this in the number of patients who have it, but it is currently considered a rare disease. And so rare diseases are diseases that in North America, there are fewer than 200,000 Americans that are affected by it. So every country has their own slightly different definition of them, but that is our definition. And so you can imagine if you have symptoms and you go to your physician and they can't diagnose. And again, this comes back to me coming out of college and saying there are gaps in our knowledge, and I don't want to be sitting in a room with a patient and not have the answer for them. Physicians don't have all the answers because we haven't given them all the answers yet through the science. And so this is something that I was naive to and really felt like my eyes were opened to, and it changed my perspective of the least of these. And so this gentleman played a big role in that. So again, a lot of our data is applicable to hypereosinophilic syndromes, but we didn't study the syndrome specifically. This is a group of different disorders that all have some shared characterizations or characteristics, but they are different entities, exceptionally rare. So our treatment of these diseases now are significantly better. In the 70s, though, you only had a 12% chance of greater than 3-year survival if you were diagnosed with a hypereosinophilic syndrome. And it is much, much, much better today. We still have a long ways to go. Well, this gentleman, my second or third day in the lab, he walks in with Peter Weller, and Peter said, here you go, can you draw his blood, please? Okay, he wasn't on the schedule, and it was because he's a hypereosinophilic patient, and so every time he was anywhere near Boston, he came in to give a sample, which was just amazing. So getting to talk to this man and hear about his story and understand what a phenomenal patient advocate he became and how much money he raised to understand this disease that absolutely changed the landscape of how this is understood. And so meeting folks like that, it really opened my eyes. Coming out of Gordon, I thought I had to go on a medical missions trip if I needed to find the unmet needs, and they're in our backyards as well as overseas. And so just kind of keeping your eyes open and your heart open to where God is showing you where he wants to use you or us on these processes. So I love this cell, and so I'm very happy to tell you it is not all bad. It does good things. And so I had, in one version of this talk, I had shown a CBC count because I thought we all get blood work done. We can all read those, and it's a good way to relate. In a North American healthy CBC, you might have 0 to 0.5 times 10 to the 9th eosinophils in your blood. So it's pretty close to 0. That's because the blood is a highway. They're not living there. They're just transiting, and they're going to tissues. And so now that we have better ways to detect cells in tissues, better ways to quantify, and frankly, new to look, we now understand that eosinophils seed a number of tissues at baseline in healthy people. And so they're shown here, the metabolic roles of adipose tissue eosinophils is just exploding right now. So there's so much more that we need to understand about that. There's another study, I actually didn't include it here, but eosinophils are involved in inflammaging, which is one of those concepts of more inflammation over time has some of the aging effects, and they're related to cognitive decline. And so they sewed 2 mice together, and they showed eosinophils from the young mouse crawling into the older mouse improved their cognitive function. So that's just a side note that has nothing to do with this, but it's really cool what eosinophils are finding. The whole point of this, though, is we finally realized they seed all of these different tissues. The highest density are actually in our guts. And so this was exciting, and I decided the right place to go for the next set of questions is the gut. The problem is I was 3 years into a faculty position at Harvard, and I had an R01 in the lung. And so any of you who are aware of the whole live or die by funding, once you get that R01, especially if it's your first one, you pour 120% effort into that and pray every day that you can keep that grant going, found your career, and get your next one. You don't switch organs 2 to 3 years into your very first R01. So I talked to my division chair about this. I said, this is the right question. And he looked at me and he said, it's a very interesting question. It's a very important question. That is professional suicide. And he was right. He was really right. And this is not, I'm just out of college and I might have trouble paying my rent. This is, I had a child, I was paying a mortgage, my husband's in full-time ministry, so you can guess where the breadwinning was coming from. So they're real significant decisions. But I really felt like it was the right place to go, so I did. We moved into the gut. We finished all of our obligations for the lung studies, but we certainly didn't pour ourselves into driving that next grant. We switched to the gut, and I'm still here, so clearly that worked out. We did land another R01. There were 6 months of sheer panic and terror while we were waiting for that to happen, but it did come through, and I will also say, here's how God is amazing. I just got another grant last year, and it's looking at the interaction and crosstalk between the gut and the lung. So it all comes back around. So what we know about eosinophils in the gut, and I didn't show this was in the literature, is it's everywhere, from the stomach all the way down to the rectum. The only place you don't have eosinophils in a healthy person of the gut is the esophagus. They do a lot of good things. They contribute to the barrier integrity. They maintain those epithelial cells. They shape the adaptive immune response. They contribute to metabolic homeostasis. They're playing significant roles in keeping us healthy, and we had no idea. We thought these was an inconsequential cell. The problem is there are some gastrointestinal inflammatory diseases where eosinophils just kind of come flying in, and they tend to be bad, or we think they're probably bad. So eosinophilic GI disorders is the big one that I'm focusing on in my role now. And so I mentioned eosinophilic esophagitis is one of those rare diseases. The eosinophilic GI diseases are a whole spectrum. So depending on which level of the GI tract you have those extra EOs, they get a separate name. And what's important about this is they're probably nuances in those different diseases, and we can't even study them as rare diseases unless we're all talking the same language. And they didn't have ICD codes, which is what physicians use to put it in the system of what the diagnosis is, until later in the 90s, and now these EOG, EON, EOC, all of those separate entities are even newer than that within the last few years. And so it's really a new field. So we're asking these questions without all of the pieces in place. We also know that eosinophils are important in inflammatory bowel diseases. That's Crohn's disease and ulcerative colitis. And so we have some projects in the lab now really looking at this. Again, we think they're bad in most cases. There's some data that says they're helping things along. So we really need to figure out who's bad, who's good, and what the difference is. Colorectal cancers is actually the one place, generally in solid tumors of the gut, if you have eosinophils there, it's a positive prognostic indicator. So they're likely doing something good in that realm. So this is the muck that I stepped into to try to figure out our next question, which is this. Are they friends, are they foes, or are they irrelevant bystanders? So the answer is yes. The working model that we have is we think that there are at least 3 types. There are probably more than this. And again, most of this is unpublished. Some is coming out from other groups where they are also finding similar things. I have a slightly different take on this, so depending on who you read, we'll see who's right at the end of the day. I see at least 3. So I see one that is what you have in your gut, your normal conditions. Those are healthy, homeostatic, eosinophils doing good things. We also see a different type of cell coming in under these disease cases that look inflammatory. And so those are the ones that are spewing their granules in that picture. And I also think we see a repair, which seems to be a little bit in between the two. So I think they're going to have even more functions than we really understand. And I think at the end of the day, if you have more of the good homeostatic ones, you are tipping that balance toward health. You're doing good things. If you get too many of those inflammatory ones flying in, now you're tipping the balance the other way, and now you're going to have a disease problem. I also think the resolving eosinophils can help to reshape that and re-equilibrate the balance. This is what we're working on. This is our paradigm and is the question du jour. And if you were in the molecular biology class today, you played with transcriptome data sets here, because this is where we think about these concepts that came out 20, 30, 40 years ago to understand that a cell is going to express a certain group of genes. And that cell could walk into a different environment, be the same cell, but it just went into a different environment. It's going to express a different set of those genes. And so looking at the transcriptome to be able to get a sense of which pattern and profile the cell is expressing is giving us a picture of the different subtypes of these cells. Now, I'm a protein person, so I am looking for the protein receptors on the membrane because I want something I can stain in a biopsy that I don't have to digest the tissue and pay $200 a sample to do RNA sequencing. But the concepts are there, and we can learn from each other and really find those receptors. So this is my question when this man started calling. So this is Dr. Furuta. He trained at Harvard and then was recruited to University of Colorado School of Medicine and Children's Hospital. He was studying eosinophilic esophagitis and treating patients here and really wanted to create a program that was holistic so that he's a gastroenterologist. This is a GI disease, but it's a food allergen-driven GI disease. So you also need an allergist. And so he has on his team, he's pulled allergists together with GI docs. You also need nutritionists because these are kids who need to not fall off the growth curve when we take all their foods away. You need your social workers because this is a really devastating family disease in some cases. And you need the nursing staff and the dietitians, and you need all of those people in one place. And he created that program in 2007 at Children's Hospital, Colorado. And so in late 2017, he called, and he also does research. I mean, the man's a little bit intimidating. He gave me a call and he said, I love this program. This is what we've done. We're doing so much good. I have the best group of people. And he's right. It's really an incredible group of people. He said, the next step is I want a global footprint to really move the needle on this disease, and we have to have the research. Do you remember that early slide where before you could do anything over here with the medicines, you have to ask the basic science questions? He got that. And so he said, we want to be innovative. We want to lead the charge on this. Can you come and can you direct the science piece of this so we can partner up and get to that point? And that's what sold me on his program. It took him 1.5 years to sell me, but it is what eventually sold me. I am going to take a minute, though, and this is a patient video that I'm going to show. This was shot before I came. It was stories like this that helped really get me on board, because as I was working through everything, I love collaboration. In case you didn't notice that from all of the people on there, I love collaboration and I love team science. And so looking at this video, just watch for those aspects of collaboration and team science. Also remember, I went into this because I was frustrated about the gaps. I didn't want to be a physician sitting with a patient and not having the answer for every patient that came through the door. And I love that people are so talented and can practice the arts of medicine. That wasn't me. So I really needed those hard science pieces in there. So listen to this mom when she talks about going to pediatrician after pediatrician, because we don't have all the information and the tools yet. But there's another piece of it too, and this comes out from, this is a relatively early disease, that were recognized disease, although it's been around for a long time. So we don't have all the epidemiological data. The first paper that came out describing the epidemiological background here suggested this was primarily a disease affecting Caucasian males. The male part is true, the Caucasian part is not. So as you listen to this and hear what it takes to get a diagnosis, what it takes to treat the disease, think about what this means for people who don't live in an urban center where they can have a sedated endoscopy. Someone who is already struggling with food insecurity, and then you tell them your child can't eat all of these different things. And then you give them the list in English, and that's not their first language, and I can barely read ingredients on some of these packages, but now you add that other level into it. So really think about what this means for the least of these, and how do we make sure that we're answering these questions well. So here's the video, if it plays. Oh, good.
Kyla has a disorder called eosinophilic esophagitis. We call it EOE for short in our household.
Much easier.
We probably noticed with Kyla that there was something off with her when we introduced solid foods for the first time. She went from a pretty happy baby to really fussy, didn't sleep, wasn't gaining weight well, and we talked a lot to pediatricians about it who dismissed it kind of as first-time mom, you're worrying too much, she's fine, just let her adjust. She started to fall off of her growth curve into her toddler years. It wasn't until she was in kindergarten where we finally decided to give her an endoscopy for the first time. The visual of her esophagus was terrible. It looked like a cat had scratched up and down her esophagus. It looked like there were rings where it was stricturing all up and down. And that was when we got the diagnosis of eosinophilic esophagitis, which we had no idea what that meant. So we were with a GI doctor who gave us this diagnosis, and she said there's a component of this which is an allergy, so I need you to see an allergist. So I went and took her to an allergist. His opinion was EOE was not a food-based disease, and she could just take some Zyrtec and she would be fine. And so I had two separate, completely separate treatment choices, I guess. But I went to her pediatrician in tears saying, I don't know what to do. Doctors are telling me completely separate things.
I think we were kind of like, just help us in some sort of way.
It was really frustrating because you think that doctors have the answers all the time.
And she said, you know what, maybe a second opinion would help you. And I was like, please. She sent us to Children's, which kind of started us on that trajectory towards where Kyla is now. When you finally do get a diagnosis and a doctor tells you, well, it's the food you've been feeding her that is making her sick, you just feel horrible. You feel like a terrible parent to know that what you were doing all along that you thought was helping your child was really hurting them.
Yeah, there was a lot of guilt, I would say, on our end.
If the child's gone undiagnosed, we first address the parent's feelings of guilt and anger You can imagine if you have a developing child and you can't feed them, how devastating that can be for a mother when feeding a child is a key component to parenting.
There are all these different factors that I think it's really not something that somebody did wrong. I think the important point is recognizing that this is something my child has, and how do we move forward in a way here that will provide the best outcome.
The best, by far, part of being a part of Children's is the collaboration between the doctors. We can go one visit and see an allergist and a GI doctor and a child psychologist and a dietitian and a feeding therapist.
We have a social worker, we have nurses that are skilled in teaching people how to take care of the disease, and we all talk together.
We had weekly team meetings and we discuss our patients that we just saw. So not only are we seeing them all together, but also in that meeting we're discussing what our next steps are.
The other part of things that I think is really important about thinking about how we empathize with the families and with the patients is what are the barriers to care? And one of those is related to endoscopic procedures. So having an endoscopy requires being put to sleep for anesthesia, which can be quite traumatizing.
It was tough to watch. No one can ever prepare seeing their kid being put under anesthesia, but in the back of your mind, you know, it's for the best.
There are a number of innovative aspects of the program that have been really pushed forward by the research that's been done here.
And I think one of the things that makes our program special is a transnasal endoscopy. And this is a 6 to 8 minute procedure that children can do while they're awake.
This nasal endoscopy is a life changer. I mean, it's great. In and out real quick, no anesthesia.
The tube goes down your nose and it goes into your throat, and then they take the biopsies that way. You wear VR goggles and you can watch a movie in it. It makes it go a lot faster too.
Treatments for eosinophilic diseases, either medication management or dietary management. We wanted to find out specifically what they would like to have done, how they would like to have this treated so that she could live the best life that she could.
The doctors have been so supportive of our personal choice that we would like to limit medication. We did not want to go down the path of controlling it by medication if we could control it by diet.
So as far as the dietitian's role in that process is, we're part of the education team that can really lay out the different treatment options. We're there for taking foods out, reintroduction of foods, and just kind of managing their everyday life around food.
Taryn and I try everything that we're recommending, so we want to make sure they're good alternatives and we're making good recommendations that the kids will be happy with.
It's nice to have someone that's alongside you. You want someone alongside you to help build you up and to help understand what you're going through, and that helps you move forward yourself. So that relationship aspect is very important to us and has helped us to get where we are right now.
Unfortunately, it is a long-lasting disorder or disease process.
Everybody's been dealt a hand in their life. Other people have things that might be a little more difficult than what we have to deal with.
You can empathize with your child to a certain extent, and then you need to help move them forward.
It was a nice resource to have someone else say, you know what, you're right, this isn't fair, but this is your life, and you can either choose to sit and complain about it, or you can rise above it. It would be foolish to say that this is not a family diagnosis, because it is.
Her younger brothers, they don't understand quite what's going on and don't always like the menu options.
We try to make one meal Kyla safe, but there are times that they're having waffles in the morning that aren't food that she can eat, and that's okay. So as much as it feels like at the beginning, oh my gosh, we're never gonna live a regular life, you adapt.
It's really refreshing to see families I've seen for a long time. After a few years, they're like, oh, we're old hands at this. We don't even think about it anymore. This is just how we live, you know? And they really don't. It's just been kind of woven into the fabric of their life.
It is so satisfying to be able to develop a relationship with the patient and family when they come in.
And it's so fun to be able to see a patient at diagnosis, maybe as an infant, early toddlers, and get to kind of follow them through the different stages and progression through life.
I think it's amazing to see the children get more confident, but then the parents get so much more confident, and you can see their stress and anxiety levels go down.
And I can speak to the moms who are newly diagnosed, who are standing in the grocery store crying, because I was that mom at one point where everything you pick up is a no. It feels like it's never gonna be normal, but it does. It just takes time. We tell her that, you have eosinophilic esophagitis, but that's just a tiny piece of who you are. She's just a normal kid, and you can still do whatever everybody else is doing.
My wish for the patients that I'm working with is over time to really help educate and build their confidence so that one day they don't need us anymore.
I think the other part of things that brings great satisfaction is seeing the field itself progress and help us to provide better care. There's a lot of hope, and that's something that can't be underestimated. I think that we've come so far in the last decade.
And there is community out there, which I think is really important, to try and find other people who really do understand that this is hard at the beginning. It's really hard, and day to day can still be difficult.
Just because you have EOE, it's not your whole life. You're still a normal kid, and you can do it, and you will definitely get through it.
So sorry a little bit for the PR plug, but it's the only way you can get permission to use the video. So I think you probably noticed some of the points in there, just of there are so many unmet needs, and just opening our eyes to kind of see who's in our backyard, following obediently where God is leading us, we're going to be able to answer the questions that he's bringing us to answer. I had never heard of this disease when I first started, so I would not have known to kind of even look into this. I won't spend time on this because I don't think it's most people's interest, but just to give you context, these are pictures of an esophagus. So on the far, it's my left, is it your left? Yes, your left. That's a normal looking esophagus. So the food's gotta go down there, right? So you need enough space for that food to go through. When the mom talked about structures and rings, that's what that kind of center picture is. And if you notice that hole for the food is a little bit smaller, right? So if that gets smaller and smaller, you can imagine that becomes difficult. And if food is actually getting stuck, a bolus of food being stuck in the esophagus, that's an ER visit for these kids to have it removed. And adults as well. This is not a pediatric disease per se, it's both adults and kids. So we're focused on kids right here, but it's not limited to kids. And if you look at this biopsy, so when they go ahead and put that tube down, and they'll take tiny, tiny, tiny little bites, and they're just tiny. So when the scientists ask you for a couple extra biopsies, super, super ridiculously tiny, please say yes, if that's okay. There's my plug. So there's a biopsy on the far right. If you can appreciate it at that magnification, I'm not sure, those really bright pink, I know the tissue is kind of pink, but there are really bright pink cells. There are your eosin-loving eosinophils with their H&E stain. And the point I want to kind of make from this slide, though, is we have some things we can do. I say we, I'm not a physician. There are things that the physicians can do, like dilations. Steroids are often the first line to get the inflammation down, but it's very little. It's a food allergen-driven disease, but you can't just have a prick test because it's not an IgE-mediated disease. So we don't have a test to know what the food allergen trigger is. We're working on it. We have a project on it. I think we have maybe the answer in mind, but it's not here yet. And so for these kids, you take a lot of foods away, and then you wait 6 to 8 weeks. You sedate them and do another endoscopy, and then you start adding them back one at a time, with every 6 to 8 weeks doing another sedated endoscopy to see which food is actually the trigger. Like, this can take 1.5 years, and this is a lot, especially for kids. So there's no cure, and so that's there. But the first medication actually received FDA approval actually last month. I don't think it's going to solve everything, but I bring it up because it shows you how contemporary it is, and also, guess what that medication is targeting? IL-4. So it all comes full circle. So this is our program now. I have joined, and I started at the very end of 2018. And we've organized, the vision that we're casting now is really looking at the research half of it. This is just the research piece. This is not the clinical piece. Just the research piece of it, we really have it split into 3 levels. My heart is in the basic and the translational steps. That's the blue and the yellow there. So my projects are there. And I should point out the mucosal organ crosstalk is the lung and gut crosstalk grant. The looking at Notch 2, that's a project actually Dr. Cornwell worked on. You probably saw Courtney's honors thesis was to make the mice where we knocked Notch 2 just out of the eosinophils. So these are Gordon students who are playing in all of these projects and getting publications. We have a new paper that's going out the door that took us a little while to figure out what was actually going on, but it's ready to go now, and Courtney's a co-first author on that. So Gordon students have really paved the way here as well. So we have the basic studies, driving hope for tomorrow. We're bringing it directly back to the bedside with the translational studies, and we're using patient samples to really keep these human-focused along with our mouse models. But the other piece that has been fun for me that I haven't had in the past that was part of what also attracted me to trying this new challenge is that top pink box. We focus on, as scientists, we want to cure things. But we forget that there are people living in the space between now and the cure. And so I love that there are clinical scientists on this team who want to ask those questions. So they talked about the transnasal endoscopy that you saw, the tube going up her nose. That's the far right over there. It's game-changing for a lot of these kids who have to have multiple endoscopies. The questions about socioeconomic status, this person up here on the left, top left, Dr. Mehta, she's a new physician really trying to get herself established, and that's where her heart is. She's pouring herself into research to try to figure out what are the barriers to care. And so finding ways to really focus on these questions as well. We sit in on all the clinical meetings. They talked about we all meet once a week to talk about patients. We have the scientists sit in on those meetings too, because the nurses talk about things like every patient asks if they can at least bake the egg and stick it back in the diet or not. Like, we aren't even aware of some of these questions until we sit in the room with the folks who are on the front line and getting those real world data points in, because we can ask that question. We can bake our antigens and see if they're still antigenic in the lab, and then we can come back and tell you. So there are things that we can do when we really work together. So I'll just leave you with this. This is my view of what the journey looks like. If you're a basic scientist with a kingdom focus, we are initiated by compassion. And that compassion is really, it's seeing the people that I feel like God is opening our eyes to see. And I think that's different for different people in different times, and we're seeing the right needs at the right times. We're inspired by worship. And if we carry all of this out in a faithful exploration, and there are a lot of scary, risky steps during that faithful exploration, we will be used as this conduit of blessing and hope, and we can take part in this bigger picture. So I will leave you with that. Thank you so much.
That was wonderful. Oh, I love that. I'm teaching immunology right now, so I was just like resonating with some of that stuff.
Thank you.
We have time for some questions, and I'll bring the mic over to you. Does anyone have a question? Let me just begin with maybe the first question that popped in my mind as you were closing was, do the patients with the EOE, the esophageal eosinophilia or whatever, do they all have similar foods that cause it, or is it different for every patient?
No, that's a great question. And I'm gonna just preface this with, I'll answer that one, but if they're too clinical, we'll see. It's a great question. So we actually have 8 foods that tend to be the most problematic. And so they have things set up for an 8-food elimination diet, an 8-fed, or a 6-food elimination, or sometimes there's a 4 and a 2 depending on your place. There's also a 1-food, because the number 1 is milk.
Eggs. Oh, milk.
Milk, yes. So the different ones in different areas are slightly different, but milk is the big one. There's egg, there's wheat, there's soy. So they're kind of the traditional foods, but it's not, I haven't seen a peanut EOE, for example. So it is interesting that it seems to be a little bit of a different antigenic spread. But milk solves the problem in a nice proportion of patients.
Wow, that's really fascinating. And I have more questions about FC receptors, but I'll save those.
You can go with FC receptors. We're good.
Oh, do they have IgG? Is it stimulating? Oh, we'll get into it later. You guys, who has a question? Yeah. All right, here we go. Dr. Dorothy Borse.
Yeah, I was following your description of the IL-4 back when they were smothering the worms and trapping them with DNA. And then, so I got a very positive view. And then several times you talked about the positives and negatives. And I couldn't always tell whether it was just an overactivity of the eosinophils when it was problematic, or too many in number, or they're all triggering to let their DNA out easier, or like what makes the difference between being so positive with these worms and then being a mixture of negative and positive?
Yeah, that's a great question. So interleukin-4 is tricky. It's a kind of a classic type 2 cytokine. So you need it, right? If you want a type 2 response against a worm, it's a good thing. Eosinophils make a little bit of it compared to, say, a T cell. So a T cell is going to, if it's a type 2 C or Th2 T cell, it's going to get activated and it is just going to drop gobs of IL-4 that's going to go systemically and have effects. Eosinophils carried around in little bags. They can also make it, so they can either replenish their bags or secrete it. But it's a much smaller little dose in the right place at the right time. And so that's a much more complicated question than it actually seems to be. So the cytokine itself does good things, and if it does too much of a good thing, it's a bad thing. And if it does the good thing at the wrong time, it's also a bad thing. So it's all context dependent, and then which cell is secreting it and where, and therefore how far it can go, also changes the story. So it's a really complicated question to ask. I think what you're asking is, why is the Dupixent that's in IL-4 targeting therapy working here? And I think the IL-4 that's being targeted here is probably predominantly from a type 2 Th2 cell, a T cell. It's probably not the eosinophil-derived IL-4. It could be. We can't, I mean, it's blocking all of it, so it could have any of those effects. But it's different points of the different immune responses will need different amounts in different localizations of that IL-4. So in the parasite infection, eosinophils were secreting tiny little amounts to a particular cell type, the macrophages, and telling them to do something. T cells that marched in were dumping buckets, and that was going around the body, and they were also dumping buckets of IL-5, and the IL-5 is going to the bone marrow, and it's telling the bone marrow to make more eosinophils. Then the eosinophils are running in. So they're all really complicated and interconnected. Does that make it muddier or answer your question?
No, I think that was pretty helpful, and it actually raised a second question, a follow-up question, because we all heard a lot about cytokine storms in COVID. Is this related to that at all?
I think the concept that anything that you turn on to fix something has to also be turned off is a concept across the board. And so cytokine storms are scary because, again, it was induced to kill the virus and to solve the problem. And then if the message doesn't get to the cells that the threat has been neutralized, so now stop, then that's a problem, I think, in any disease setting.
Dr. Zheng had a question.
Yeah. And I'm curious about the bad eosinophil cells, and you illustrate the cellular.
Nope, just hold it to your mouth.
Oops, okay. So those cells will shoot out the granules on the left, and eventually the nucleus is bursted. Bursting the nucleus. So what happened?
Splatting.
Yeah. So what happened at the tissue or organ level? What happens to the DNA traps? Yeah, I'm curious to know a little bit more there.
So it depends. So there are a couple of things there. Number one, we see cytolysis, which is the process, on a lot of different patient biopsies, but we don't do EM to see the traps in all those biopsies. So a lot of that we don't know yet, but it also depends on where. So chronic rhinosinusitis also has traps. It's both neutrophils and eosinophils. And so the consistency of those different traps can be different based on which cells spit them out. So even thinking about, did your trap come from a neutrophil or did it come from an eosinophil? Is it an enclosed area and clogging up your sinuses, in which case now we really have a problem of all this goo? Or is it just in a tissue that has enough macrophages and other things that are going to be cleaning up all the mess more quickly so they can do their job, grab the bacteria, and then be cleared? It depends on what tissue and in what time. So what we don't know, or at least I don't know, and I don't think it's known in the literature yet, is how long do those traps stay there? Like that, we don't know yet. We'd have to do EM over multiple progressions of probably a mouse model, because I doubt a patient would want to come back for every 10-minute biopsies, but.
Other questions? Dr. Camp.
Translation. You talked about it in several places. You're interested in translation, the mechanism in terms of the disease. Do you want to say anything about some aspect of translation that you're working on? Some, you know.
Do you mean like basic research versus translational research?
Translational research.
Yeah, absolutely. Yeah. I mean, we could even, we could hit your FC question on the translational research. We think that, and there's a lot of data, and we've published on this as well, the allergens or antigens that are driving EOE are likely recognized by IgG4 in the tissue. So Dr. Camp, as an immunologist, is now hearing, wait a minute, IgG4 is supposed to be that isotype of antibody that turns off food allergy. IgE is bad, and we try to treat IgE allergies by giving low doses of the allergen in patients so that they can upregulate their IgG4 and balance that out. So it's supposed to be a good thing. Apparently not always. And so we don't know if it's part of the pathology or just a symptom of it, but not only the levels of IgG4 in circulation seem to go up a little bit, but the levels of IgG4 in the tissue really elevated in EOE and eosinophilic gastritis. We just published gastritis and duodenitis is also elevated, and that came out just this year from our work. The fact that it's upregulated in the esophagus has been known for about 1.5 years now. So I think what we're looking at is the body is trying to not have an IgE-mediated disease. I'm making this up. We don't have data to support this yet, but this is the hypothesis. I should probably clarify that. I think this is part of this process of not generating your IgE-mediated effect, which is good because IgE causes anaphylaxis. So that's game over, right? So it's good that we're skewing it away from that. I think this is a negative outcome of that. I think if you accumulate too much IgG4 in a single spot, it may be part of the whole disease process. So one of those translational projects is we're looking at the IgG4 in the tissue, and we're asking what the antigen specificity of it is, and we're wondering if we take your biopsy at diagnosis, and we digest it, and we figure out which antigens your IgG4 is specific to, which of those 8, if it reacts with egg and milk, but not the other ones, is that enough to say, okay, those are your triggers? I mean, maybe not, because maybe you had to have eaten a certain quantity of it before I mean, there are lots of different things that we're trying to play with, but that is my hope for that project, is that we're gonna be able to say, from your first biopsy, we can digest it, we can figure out what the antigen is, and we can tell you, take these out of your diet. But we'll see.
Cool.
Students? Simplistic. We want simplistic questions.
When you say milk and eggs, is it like other allergens that you can't have even a trace of it, or is it just not drinking milk?
That's beautiful. My guess is the threshold is a little bit higher, so you can take in more of the allergen. However, it's tricky because this is not, so for IgE, when we try to see if a kid, again, when the physicians try to see if a kid is going to react to something and they want to add that food back, you have them in the clinic and you're watching them because you know if there's a problem if they start having the physical manifestations of that. These diseases do not have that kind of an immediate response because they're not IgE mediated. So you're not cross-linking your mast cells and blowing them up. So for this, you may not know right away. So it's really hard to know how much has been ingested in that time. And then sometimes the kids feel better, but the inflammation is still there because they're still having some of that milk or whatever. And the problem with this disease is, again, we don't know for sure yet, but the data is very, very strong that you get chronic inflammation over time that eventually leads to the strictures. And that we don't know if it can be reversed. So not treating that inflammation because you don't really have the symptoms because they got a little bit better, that gets tricky too. So yeah, so they're tricky questions, not so simple.
Our next chapter in immunology is hypersensitivities, which includes allergies. As you mentioned, usually we think of allergy as IgE mediated, which is this kind of antibody that causes, sudden death, can't breathe, anaphylaxis. But you're talking about IgG, a different kind of antibody that is triggering perhaps these eosinophils or in some way. So in the textbook, there's 4 categories of hypersensitivities. Is this a 5th one? Do we need a 5th one? Because poison ivy, right, is caused by T cells reacting to, the way T cells react in the tissue, and then other cells come in and do the damage. But this sounds like a really legitimate 5th or a different whole category of hypersensitivity. But my question is, do you guys refer to it as an allergy still? Yeah.
I do, but you heard on the video, some of the physicians say it's not food allergen driven.
Right.
So I don't know that it's a 5th type of allergy. We also don't know that the IgG4 is actually pathologic.
It's so the mechanism is still kind of
The
unknown.
mechanism's still unknown.
Okay,
We see it in
fair
the
enough.
tissue. I think, so we published a paper eons ago asking a very different question, but it's relevant here. We asked a question in mice. You have all of these eosinophils inside your tissue. We had found with human cells that eosinophils can express MHC class II and can actually present antigen to T cells and can activate them. And I
They don't know what MHC class II is.
We are gonna have like a, anyway.
So we don't know the mechanism really of these.
No, but this is a cool paper. So we wanted to ask the question, if you eat something, is the food that's in the lumen, if you're allergic to it, can the eosinophil in the tissue get at that food? Because
Yes.
they're supposed to be separate, right? There's a whole
Barrier
epithelial
between your
barrier
food
there.
and your immune system, right?
We're gonna talk about this in your class tomorrow.
Yeah.
actually.
But don't tell me they reach through and they get the allergen.
I thought they did. So they sit there and they do reach through a little bit, but that's not how they grab the antigen. I was really disappointed. They get it, not if the mouse is naive, but if it's an allergic mouse, they can get the allergen or the antigen. And it turns out that you can passively immunize. I'm saying this just for Dr. Camp. You could passively immunize these mice by taking the immunoglobulin or the IgG from a sensitized allergic mouse and give it to the other mouse. And now all of a sudden, the eosinophils in the gut can get the allergen. And so the point that makes that so interesting is we tried it with IgE and it didn't work, where we took the IgE away and it didn't do anything. But we could transfer it with IgG. So I think that eosinophils getting antigen is an IgG-mediated phenomenon. So maybe we'll rewrite the textbook.
FcRn, I cloned it. That's my gene. That's the one. All right, so any final questions? We have time for like one more question. Dr. Camp.
I want to know if you had one experiment you did in your whole history of doing science that really, wow, this has got to be one of the greatest results, the sort of that barnstorming thing that really, I'm continuing on. This is great. Can you remember an experiment like that?
I have one that I will tell you off camera because we haven't published it yet. But
One that you've
I
published.
think this is when the free granules that got thrown out of an eosinophil and sat in the tissue, when we found receptors on the granules facing out. And then we stimulated those receptors. So there's we showed differential secretion of all of those different cytokines in the granule. We showed we could stimulate with different things and make different profiles or patterns of those cytokines come out, which is cool. But those granules sitting in the tissue, I stimulated them with the same group of different stimuli, and they secreted just as differentially as the cell itself. So the fact that this teeny little granule with a little city inside it of tubule vesicular structures that ebb and flow, they can function in that kind of level of regulation blew my mind. So that was my moment.
Does that mean that the granules normally are secreted and then are stimulated to specifically secrete what they're supposed to secrete?
They can. I don't know if they do in vivo. Like I don't know in vivo how long they sit there. It's the same.
Yeah,
question
maybe
that we know
that's
in
the normal way it works.
But it does when we do it experimentally.
Wow. Oh my goodness, you guys. Let's give a round of applause for Dr. Spencer. Thank you.
Yes, thank you. Thank you, Lisa. This has been wonderful for this political scientist to see that they have fun in other parts of the. I really got a sense of the amazement of the wonder of what you do, and it was beautiful to see. So thank you. Thank you also to those who were joined us on the live stream. This was being streamed at the same time, so forgot to mention that earlier. Thank you. But thank you all of you for coming. Lisa will be here for a few minutes for you if there are still some lingering questions. But thank you so much for coming. It's been a pleasure to have you here. Have a good evening.
Thank you all.